epigenetic modifications. There are three different types of the modification which are considered as epigenetic modification. First one is methylation of the cytosines. Second one is modification to the histones which are present in the chromosomes. And third one is siRNAs, the small RNAs which are present in the nucleus. If we see that how modification is done at a cytosine or how methylation of the cytosine is done, a methyl group is attached with the help of an enzyme. The name of that enzyme is methyl transferase. So this is the structure of the cytosine. This is carbon number 1, carbon number 1, carbon number 2, carbon number 3, 4 and 5. So our enzyme, the name of that enzyme is methyl transferase. Methyl transferase add a methyl group at carbon number 5 of the cytosine. So this is carbon number 1, 2, 3, 4 and 5. So at carbon number 5 of the cytosine, a methyl group which is CH3 is at the position of carbon number 5. This phenomena is called as cytosine methylation. What are the effects of the cytosine methylation? Genes can be switched on and genes can be switched off due to cytosine methylation. If we see here that this is the cytosine and a methyl group is attached at carbon number 5, so this cytosine is methylated. While in case of histones, these are the histone modifications, phosphorylation and acetylation are the phenomena which are added to the histone modifications. So these two phenomena, acetylation and phosphorylation, modify the histones. What are the effects of methylation of the cytosines and modification of the histones? If we see that genes, they can be switched on and genes, they can be switched off. If the cytosines are unmethylated, then the gene will be switched on. Here we can see that these are unmethylated cytosine. Cytosines are not methylated. Or in other words, we can say that if there are acetylated histones, then genes, they will be switched on. While on the other hand, genes will be switched off if the cytosines, they will be methylated. So these are the cytosines. Here we can see which are present on the DNA structure. These cytosines, they are methylated or deacetylated histones. So these are the deacetylated histones. So genes can be switched on and genes can be switched off due to epigenetic modifications, and epigenetic modifications can be DNA methylation and it can be phosphorylation, acetylation of the histone molecules. In this diagram, we can see that methylated cytosine is present in CGP islands. These are called as CGP islands. So CGP islands are those locations on the sequence of the DNA where cytosine is converted into 5-methyl cytosine. So those regions or those sequences are called as CPG islands. So as a result of CPG islands, there are certain functions. They are the compaction of the chromatin, subnuclear location, gene transcription, RNA metabolism, RNA replication, and transposons silencing. So these are the phenomena which are affected due to the CPG islands. And at the organism level, there are certain phenomena that appear. Those phenomena are called as genomic imprinting, changes in the development, cellular pluripotency, and proliferation and differentiation. So these are all phenomena at the level of organism which are due to CPG islands, And CPG islands are those regions in the DNA sequence where cytosines they are methylated. In this diagram, we can see that how a cytosine is methylated into methyl cytosine, and this is the methyl cytosine which is already present in the DNA sequence, and de novo methylation of the cytosines can also occur. So here we can see that two of the cytosines they are now methylated. Cytosine acetylation occurs at the regions which are called as upstream sequences. Upstream sequences of the gene are those sequences where there are promoter regions. Histone modifications. Histones are the proteins which are present in the chromosomes. Most of these histones they are basic in nature and have the positive charge, while on the other hand DNA has the negative charge. So if the histones they are acetylated, it means the genes they are functional, and the vice versa. In this diagram, we can see that what is the difference between the acetylated and deacetylated. These are the histones which are acetylated, in which the compactness of the nucleosomes have been released. They are far away from each other, while on the other hand, these are the nucleosomes which are compact and placed side by side. So there is histone acyl transferase that converts the compactness of the histones into a loose form. So this is difference. So this part of the gene will be switched on. It will be switched on and it will be switched off. Histone modifications mostly occur on the extended ends of the histones. In this diagram, we can see that how the acetylations occur on the histones. These are the histones, and they have branches. This is the branch of the histone. This is the second branch of histone H2B. This is the branch of histone H2A, and this is the branch of histone H3. On the branches of the histones, we can see that there is acetylations. These are the acetylations which are present on the branches of the histones. Histone acetylation is due to two enzymes. One of the enzyme is HAT, histone acetyl transferase, while the second enzyme is HDAC, that is histone deacetylase. Higher the level of the acetylation, it means the gene will be more expressed. Epigenetic modifications can also occur due to small RNAs. Those RNAs they are called as siRNAs. We can conclude that there are three different types of the modification in the chromosomes or in the DNA. They are cytosine methylation, histone modifications, as well as siRNAs.